Good morning. We are here in the final day of SOHO, and this is the best of SOHO. And I guess one of the best sessions, because one of the questions that is remaining unanswered in CML is when to stop therapy. And here with me are two distinguished professors in CML who have tried to answer this question in a very lively debate. To my left is Dr. Deininger from Huntsman Cancer Institute, Utah, and Dr. Rousselot from Versailles, France. Welcome, gentlemen.
Thank you.
Thank you.
I should start off by saying that the most important question, of course, is when to stop therapy in patients with CML. But before I get to that, it looks like from the debate, both of you agree then disagree. And I guess you presented the same sets of data to quote your case. If you don't mind, Dr. Rousselot, since you started out first.
Yes, the criteria have been derived from clinical trials. And what have been seen is that patients in deep molecular response and at very initial study, they need to be in complete molecular response. We tried to discontinue those patients if the molecular remission was durable and stable over time, so at least 2 years. And those patients have been treated for a long time before being discontinued, at least 5 years of therapy with mainly imatinib. So with this condition, we were able to demonstrate that the discontinuation was feasible and that around 50% of the patients will experience a long-term treatment-free remission.
I know you quoted studies, and you quoted your experience at your center because you have a very nice setup to follow patients when they're not on studies. Dr. Deininger, on the other hand, you wanted to temper this with a cautionary note.
Well, in my mind, the studies are very good and they're also extremely well controlled, and everybody is playing by the rules. And this is why, in my mind, why the risk is probably quite manageable. But it's not so easy to translate into a real world setting of clinical care, both in the United States and probably Europe as well. And so I'm just not sure whether we are prime time at the moment, because of us, as I've tried to show, we're not following our own guidelines, and clearly introduction of treatment-free remission into general practice would come along with a set of very stringent guidelines. And I kind of have some doubts whether we as a CML community are really able to do that. And with that in mind, I think there will be rogue discontinuation, and we'll see people showing up in ERs at some point after getting lost to a follow-up and things like that. So I'm really concerned about that.
Excellent points. I guess there is a risk-benefit value to this proposition, and you very rightfully mentioned the risk of the patient knowing, fully informed about this, and a highly vigilant physician who knows what's going on with the patient. Having said that, what are the other considerations in trying to stop this? And I think you gave a set of rules, and both of you agree that if you were to do this, let's say there's a patient, a biological question, a patient who's pregnant, and in deep molecular response, and what are the criteria that you laid out?
Yes, for pregnancy, it could be quite different, because pregnancy was not a criteria for entering clinical trials, of course. And for those patients, there is quite a limited experience. Every center has their own practice, and we have to put all those data together in order to better define which are the criteria to stop TKI with a planned pregnancy. With my own practice, we just wait to have a well-controlled disease, which means that at least major molecular response is achieved on the long term, which requires a stable response. And so we can just stop TKIs and just wait for pregnancy. But only half of the women that go to this procedure, they will succeed and they will be pregnant, in fact. And the other half have to restart therapy because they were not able to be pregnant before the molecular or at least certain genetic relapse.
And if I must add, you mentioned that patients who should not be considered are patients who have accelerated phase or have a switch therapy for prior resistance and have not attained that molecular CMR4. I guess you laid out those criteria. Those patients should not even be considered for on this observational basis. On the other hand, you made a point saying that one of the reasons why we are not there is because the testing that we do, and you laid out the Goldberg Journal of Oncologic Practice and how we are not, as physicians, not adherent both in academia and also in the community. And in terms of the testing, what are the steps that we can do to standardize those testing?
Well, that's a very good question, because we've been working on that, but apparently not very successfully. As you know very well, over the years, a lot of money has been spent on education, but in real practice, I think we are far from where we ought to be. So the answer is, I don't really know. I think I guess the biggest problem is that oncology and hematology oncology is being transformed by genetics, and there is so much information coming the way of practitioners in the clinic that it is extremely hard to really follow through on all these different requirements. So I guess we have to find revolutionarily different ways of communicating that, and maybe making it much easier to send samples for testing, involving the patients more directly in the monitoring process. I think all in all, we just haven't been very successful in implementing what we actually know it's the right thing to do. And I think it's a particularly big problem in this country, where there's a lot of mobility, both geographically as well as as far as the workplace is concerned. People are switching their insurances many times, so you get different laboratories doing the test. Not every laboratory is on international scale. So I think it really is a bit of a mess. And so, as Dr. Rousselot, I think, very elegantly pointed out, I mean, they had very stringent criteria, and they really made sure that people had at least 3 tests a year at a central lab. So I think we are not really in a position to do that at the moment.
I think it is a very important point. There are 2 points that are very important: the education of the physician and also the education of the patient. From a neuroparent point of view, what I can say is that we have run multicentric studies, and so most of the physicians have been educated to do the molecular monitoring, and so they are quite well confident to do that, and it is feasible because most of the labs, they can just give the results as a standardized ratio, and the criteria for relapse is loss of major molecular response, which is very reproducible in each molecular labs. And the other point is the education of the patient. The patient have been educated because they have heard of the about the result of the clinical trial. There are patient groupment that are communicating on this topic, and so now the patients, they are able to organize the sending of the samples, but also to call the center in order to have the results within 1 or 2 weeks. And the education of both the physician and the patient, I think, will be the condition for a successful discontinuation trial and a successful discontinuation strategy for most of the patients who are filling the criteria.
Excellent points, both of you. And I know it's a very important question that we're asking, and we're trying to answer that to the best possible ways through clinical trials and also in real life practice. But I think the basic point is that you have stringent criteria, you have a very well informed patient who knows the risk benefit, and also very well a vigilant physician who knows the risks of doing such a thing. Thank you again, gentlemen.
Thank you very much.
Pleasure.